Great. So we'll record this session for those who can't be here. And of course, if you don't want to be captured by the recording, just leave your video off. All right, well, good morning or afternoon or evening, depending on where you're joining us from. And thank you all for joining us. We're really excited to welcome you to the September Mixotrophs and Mixotrophy meeting. We're here courtesy of the Ocean Carbon Biogeochemistry Working Group on Mixotrophs and Mixotrophy. And today we're going to talk about biogeochemical modeling of mixotrophs. So this builds on our July session when we talked about biogeochemical data. And the question is, okay, we have a growing and awesome set of empirical data. How do we take these data and integrate them into models in ways that balance what we know about the complex realities of mixotrophy with the tractability that we need to implement these models mathematically, especially at increasingly complex spatial scales, like global spatial scales. So today we have three absolutely incredible speakers who are going to try to answer this question in various ways using their different approaches to modeling mixotrophs. And we've organized them in terms of spatial scale of implementation. So each speaker is going to talk to you for about 15 minutes about their work, and we ask that you hold questions. Feel free to enter them into the chat if you'd like, because we'll have a panel-style Q&A at the end of all three talks. So we're going to begin at a relatively localized spatial scale with a relatively simple implementation of mixotrophy with Dr. Ken Andersen from the Center for Ocean Life at Denmark Technical University. And Ken is going to describe for us a minimalist approach to size-based modeling of mixotrophy. So Ken, take it away.
Thank you very much. Can you see my screen and hear what I'm saying?
Yes.
Perfect. Yeah, so first of all, I should say thanks for the people who are organizing this working group and this session. It's been a pleasure to be here, and I think you're really at the forefront of what's happening. So what I want to talk about here is, as Karen said, or Holly said, a minimal way of modeling mixotrophy on a global scale. Essentially, how little do we need to get some of the qualitative global features? And what I want to do is to go from a description of the single cell and the characteristics of that towards the global ocean and the global biogeochemistry. Yes, and what I want to do is to make a cell that's generic, a cell that can do everything. This is essentially an adaptation of the model that was first presented by Ben Ward a few years ago. And the idea is that I'm not trying to prescribe anything about this cell. It's a phytoplankton, a zooplankton, mixotroph. I just say it can do anything. It can take up light or do phototrophy. It can take up the salt, carbon, do phagotrophy, and it can take up the salt, the inorganic nutrients. And the carbon part of that goes into the carbon flow. Some of it is respired. The nutrient part, likewise. And then you have the biosynthesis part, where you leak surplus nutrients or carbon, which gives you the division rates. That's the essence of it. Now, of course, cells vary a lot, and to be able to generalize that to the entire plankton system, I need to describe the differences between the cells. And what I want to do is to use just the size of the cell to describe them. So, and that means I need to parameterize each of these uptake processes with respect to cell size. So we have the affinity for light, specific affinity for light as a function of the cell size or cell radius here. And there are essentially two processes that limits this affinity. First of all, is a surface limitation. That is that they prefer to put the chloroplast in the surface because the stuff in the middle will be shaded by that in the surface. Unless if cells are very small, then they can, in principle, they are so small so photons can almost pass through, in which case they're just limited by their size. This is really only relevant for the smallest cell. And the main limitation is the surface limitation here. Uptake of food or phagotrophic feeding, we don't have a general understanding of why, but the specific clearance rate for food is essentially independent of size. There's a rough rule of thumb. A cell can clear about 1 million of its own volume per day. And that's essentially this line here. That's probably limited by fluid mechanics, but we don't know exactly yet how. And the last main process is that of the uptake, the diffusive uptake of nutrients. That again is a complicated process, but we know at least that they cannot take up nutrients faster than diffusion transports the nutrients towards the cell surface. and that's essentially the limitation we have here, and that's calculated from diffusion physics. For the very small cells, there will be other limitation by the number of porters that can fit on the surface, or simply the material needed to make the porters. Again, that's a weaker limitation for the small ones. We can't see very strongly in the data, and we don't really know from first principles where it is. But just from these processes, we can actually understand a lot about the mixotrophy here. So here I've taken a cell just described by these processes here, and I put it into an environment with a certain amount of light, certain amount of dissolved organic carbon, nutrients, and a constant type of Sheldon spectrum. So, and what you see here is then the different processes. We see the uptake of light here that declines with size, the uptake of nutrients, potential uptake of nutrients, the uptake of phagotrophy. We see the smaller ones, of course, are not phagotrophs because there's nothing to eat. They would like to eat smaller stuff. But from this, if we should say just about who are mixotrophs in this case here, well, all of those that actually are phagotrophs, and since most of them do take up light to a certain degree, we could argue that all of these are essentially mixotrophs in the model, and all the smaller ones are phototrophs or pure phototrophs to various degrees. But of course, another way, a more detailed way to look at the trophic strategies is to say where they're getting the carbon and nutrient from. So here I'm saying that here they're getting a significant part of their carbon from light, potentially here more than phagotrophy. While these ones here, they actually leak nutrients. They're not getting any dissolved nutrients, and they're getting so much, so little carbon actually that it's not enough to fully fuel their metabolism. So they have a surplus of nutrients and they're leaking some. That is what we could really call zooplankton. But they still have some phototrophy nevertheless. Down here, we have the phototrophs. What we'll see in this size range here, that they're getting more carbon relatively than nutrients. So you could say that they're nutrient limited. While these ones, the smaller ones, would typically be light limited. So this is a case where I have a lot of nutrients, so that's why they are more light limited than nutrient limited. And again, this is just based on these very simple principles I showed in the beginning with the size-based scaling. And we can look at how that changes with the conditions. Just need to move this picture here. So here I have the conditions that I showed over on the left. If I change the nutrient levels, well, if there are more nutrients, we are getting more light limited. Less nutrients, I get more nutrient limited. And we have a wider pure mixotroph range here. If we change the light, we become less light limited up here, of course, more light limit below. It doesn't change the mixotrophic range so much. If I change the amount of biomass I have in the system, well, phagotrophy and mixotrophy becomes most favorable relative to the light because there's simply more food. Yeah, okay. So that's so you see, actually, just from these very simple principles of the bioenergetics of the cell and the size-based rating scaling, I get a lot of a very, say, rich system of different trophic strategies. But that's just not really a model. So what I'm doing now is taking that description I had before, saying I have a cell that can take up carbon, nutrients, etc. It can predate on smaller cells, and I put it in, make a lot of these individual cells here. So I have a complete spectrum of different cell sizes, and describe them with, this is a standard equation we will use in the biogeochemical model. We have the biomass of each size groups here. They have a growth rate, which is determined by the amount of nutrients, light, and the food from smaller cells. There's a mortality. There are a few components, but really the main component is predation that comes from the larger cells for their phagotrophy. So that one, I have an online simulation. I think I've shown to several of you before, but you can try it out if you go on this side here. And it shows the same that I showed before. For a given set of conditions, light, mixing rates, temperature, or so on, this one doesn't really work. We'll get now a size distribution. So the difference from before, where I fixed the size distribution, now that's actually a result of the model. And you see in the same way, I get the different trophic strategies, the mixotrophy, etc. And if we change the condition, for instance, if I lower light, well, I get a more wider mixotrophic range and also get fewer cells. You can see if I move back and forth here. And it scales all the way up to the ecosystem functions. So if we go on the next tab here, we'll see the ecosystem functions, the amount of energy sent to the higher trophic level, primary production, etc., etc. Just to give you an idea that just with such a simple model that actually have very few parameters, you get very rich output. And you can go one step further and put it into a global model. So here I've done it very simple using transport matrices. And that has the advantage that it's simple. It can run very conveniently, very quickly on a small laptop like the one I'm presenting on now, which is I think 5-6-year-old machine. You can run the global simulation of this model. This is the output you're getting here. Here I've looked at 3 different size groups on a global scale. And you see you get a typical pattern. You see I get too high production down here, but that's because we don't have iron and iron limitation. And you can, if I look here at 1 point on the globe, here I have at 3 different times, again, the size, and here I have the water depth. We see the size distribution. We have deep mixing in spring, and then we have a stable stratification, but the similar size distribution. And here we have the trophic strategies, just like before, with the different colors showing what is the trophic strategies, which change according to season, but also to depth. For instance, you see down here deeper, we have more dominance of phagotrophic strategies, whereas we have more phototrophic dominated strategies up in the surface. So, and all this came again, I just want to highlight again this very simple description of the cell. So this, of course, is a very primitive model. It has very little reality and very little detail and detailed strategies, but you get a lot of the dynamics of mixotrophy out of it. And that also shows that mixotrophy, well, it's something that comes out very robustly of models. So let me just round up. I mean, I think what we are doing here, and very much the people here in this working group, is to define what I call the mixotrophic paradigm. A lot of the models that has been built so far by biochemical model come from a classic NP-Sept paradigm, where we are thinking about phytoplankton and zooplankton. What we're doing now is putting mixotrophs somewhere in between that. But I actually would like to go one step further than that, because I think actually the core and the central strategy we should think about is the mixotroph. This is the obvious strategy because that's a generalist strategy, and that is usually where cells would like to would be a dominant strategy. And I think we should much rather ask, why are there actually cells that are not phototrophic? Why would they not want to get that little extra carbon that they can use to fuel their metabolism, so that they can, all the food that they get from phagotrophy, that they can use that for biosynthesis? Or conversely, why are there actually some cells that are purely phototroph and give up the lovely ability of being able to eat other cells? Well, we do understand that partly the smaller cells, of course, there's nothing to eat really. We can think of them as being partly heterotrophic, but that's more osmotrophy. So that makes sense. But there are also larger phototrophic cells. Diatoms is a great example that cannot do phagotrophy, and they have to get quite a lot of benefit of the strategy of building a silicate shell, because one of the main costs of that is saying, well, I cannot be a phagotroph, which is great. Another question, and that's a little smaller question perhaps, but a similar vein, why do some mixotrophs or phagotroph forego the ability to take the carbon from phagotrophy and put it in and use it in respiration? Many of them cannot grow in the dark. They actually need light, even though there's plenty of food. Why wouldn't they want to be able to have that pathway? So to sum up, I think the paradigm or the way of thinking of plankton and plankton modeling, I think we should go for is really to think of it as mixotrophs. They're the generalists, and what we should rather do is put in the specialists, the pure phototroph and the pure heterotrophs as the little extra bits on top of the mixotroph. which forms the core of the plankton community. So with that, I think I'll pass on. I think I did well into 14 minutes, and yeah, thanks.
Yeah, terrific. Thank you so much, Ken. And I'll give you a virtual round of applause. That was super fascinating, and I always love seeing the positioning of mixotrophs there and in such an awesome mechanistic way. So if you have questions for Ken, please hold those or drop them into the chat. And we will have a panel style discussion, a Q&A session at the end of all three talks. So we're now going to transition to a slightly different approach for incorporating mixotrophs into models, now taking into account a little bit more detail about the different flavors of mixotrophs that we commonly categorize these organisms as. And so our next speaker will be Dr. Suzana Leles, who is at the University of Southern California. And Suzana is going to speak to us about accounting for different types of mixotrophy at the regional scale. So Suzana, when you're ready, please take it away.
Thank you, Holly. Let me see, I just need to swap here. Can you all see it, my screen?
Yes, perfect.
Awesome. I won't be able to see you, but I think that's okay. All right, yeah, thank you so much for the invitation to speak to you today. I'm very excited about this topic too. And I will share with you some of the work that I did during my PhD. And I would like to acknowledge my advisors, Yor Brugman, Luca Polimene, Jerry Blackford, Kevin Flynn, and Aditi Mitra. So I think like most of you here today, or maybe all of you, I am interested in understanding how plankton drive carbon cycling in the oceans. And we know that to advance our understanding on that, we need to consider the complex metabolism of plankton and also their complex ecological interactions. And during my PhD, I was investigating mixotrophy, or more specifically, mixoplankton. So we name mixoplankton those organisms that can do both phototrophy and phagotrophy. So basically what this working group is interested about. And we know that they are very diverse, as we can see here on the left. And as you can see in the global maps, they are also ubiquitous in the oceans. But it is very challenging to identify and detect the activity of these guys in situ. So we don't really know how much mixoplankton will contribute to the total primary production or to the grazing pressure in the oceans. And then modeling studies can give us some new insights about how mixoplankton might impact trophodynamics and also nutrient cycling. Yet we still don't know much about how these different factors will affect the dominance of mixoplankton in the oceans and how their diverse strategies, or Holly was saying, the different flavors of mixotrophs can impact ecosystem functioning. So based on these, we had two questions, mainly when and where mixoplankton could dominate proteus assemblages and how diverse types of mixoplankton can impact carbon fluxes. So our approach was to model different mixoplankton functional types within a 1D model of the water column in a coastal temperate sea. So basically what we did, we start dividing mixoplankton in two groups. So in one side, we have the constitutive mixoplankton, which possess their own photosystems. And on the other side, we have the non-constitutive ones, which need to acquire plastids from their prey. So these guys, they are also known as the acquired phototrophs. So we consider these two types in our model. And among the constitutive ones, we also divide them in two different size classes to account for mixotrophic nanoflagellates and mixotrophic dinoflagellates. So this is a conceptual schematic of a constitutive mixoplankton in our model. So basically the cell will be able to photosynthesize, to take up inorganic nutrients, we respire, we release DOC back to the environment, and is also able to prey on the specific prey types. And what is interesting about this model is that since we account for variable stoichiometry, we are also considering some synergies that can happen between phototrophy and phagotrophy. So in our model, the constitutive ones, they are primarily phototrophic. In this sense, whenever their phototrophic capacity is lower than the maximum capacity to photosynthesize, then this will enable feeding. And feeding can also stimulate phototrophy by improving the nutrient quota of the cell. And when we think about a non-constitutive mixoplankton, we might start as a strict heterotrophic cell, but this cell can also acquire plastids from their phototrophic prey and then be able to fix inorganic carbon. So in our model, these guys, they are not able to photoacclimate, and also they don't take up inorganic nutrients. And here, feeding is not dependent on phototrophy. So we are mainly representing here mixotrophic oligotric ciliates. So we incorporate these three types of mixoplankton in a food web model. And this food web model is structured according to the size of the organism. So you can see here size increasing from the bottom to the top. And we also account for different trophic strategies. So we have autotrophs, heterotrophs, and mixotropes in our model. And we then couple this model with a 1D model of the water column, and we investigate how these different types of mixoplankton will impact different carbon fluxes in our model. So these are some results for our validation. Basically, we use data from a monitoring station in the Western English Channel. This is a we use 9-year long-term data sets, and we were able to represent the seasonal succession of different trophic strategies among protists. So here you can see the climatological means for the observational data represented by the dots, and the lines shows our simulations. So the top plot shows the phytoplankton biomass, the middle plot mixoplankton biomass, and then in the bottom we have the protozooplankton biomass. So after checking that our model could represent the seasonal succession of these different trophic strategies, we decided to explore how much these different strategies would contribute to the total protist biomass in our model over the seasonal cycle, and also across 2 different depths here. So first, one interesting thing is that we see that mixoplankton, they dominate during summer conditions, which is expected since it's a condition when nutrients are limiting in the water column. But we also found that they can actually be present during all periods in the seasonal cycle and also over depth. But more striking to us was their dominance just before the spring bloom. So this tells us the importance of this strategy when light is limiting in the system, and also perhaps that mixoplankton can have some implications to understand bloom initiation in temperate seas. So just to give you an idea about what our model is predicting in terms of mixotrophic strategy, here you can see the relative phototrophic growth. So from to 0 here, it means that the mixotroph is not reliant on phototrophy, and then it increases up to 1 when it's primarily purely phototrophic. So we have different lines for the different types in the model. Here we have the non-constitutive ones. In yellow, we have the mixotrophic nanoflagellates, and in green, you can see the mixotrophic dinoflagellates. So as you can see here, if we look at the yellow line, during this period, which is winter, they would rely more on a mixed strategy to grow. And while the non-constitutive ones, they are primarily phagotrophic in our model, but the importance of phototrophy increases during summer as well as during the spring bloom. And then in order to investigate how these different types impact carbon fluxes in our model, we compared our full model, which has these 3 different types, with equally versions of this model, but that accounted for only 2 types, 1 type or no type of mixoplankton. So we will look first at the gross primary productivity, and this is the results for our full model. And you can see here how GPP is partitioned among different size classes. So once we have no mixoplankton in the model, we have a shift here and now the GPP is primarily concentrated among phytoplankton and microplankton. And if we simulate only mixotrophic dinoflagellates or only non-constitutive mixoplankton, or maybe a combination of both, we still see the same pattern. So this is telling us that actually the mixotrophic nanoflagellates are driving this dynamic in our model, and they allow the accumulation of larger, they allow the higher GPP accumulating in larger cell sizes. And this is happening basically because they can control the biomass of phytophytoplankton since they are allowed to feed on this prey. We then also looked at other carbon fluxes in our model, like bacterivory, herbivory, DOC production, trophic transfer, and vertical export. And I will guide you through the results for the trophic transfer now. So basically, in the situations where we don't simulate mixotrophic nanoflagellates, we can see here that trophic transfer would decrease drastically. And this is expected from what I just showed you before. So when we don't have this type of mixoplankton in our model, we have higher biomass among phytoplankton, and this prey is not available to our top predator. And that's why we see a decrease in the trophic transfer. But what I found interesting here, and I wanted to highlight to you, is that in our runs, which we did not consider mixotrophic dinoflagellates, we can actually see a change in the direction of this effect. And actually, in the absence of this group, we have a slightly increase in the trophic transfer because this group can consume diatoms, and then will decrease the biomass of this group, which is a prey that will support the biomass of higher predators. So it's interesting that depending on the identity of your community, the impact of mixoplankton in the food web can change. If you are interested on checking the other carbon fluxes, I also invite you to check our paper. I don't think I will have time to go through this today. But basically, the take-home messages from our modeling study were that photophagotrophy can have implications to understand bloom initiation, and it is a strategy important also during light-limited conditions. Small constitutive mixoplankton, they can support higher primary productivity in larger cell sizes, and the direction and magnitude of the effect of mixoplankton in carbon fluxes will depend on the functional type in our model. Now, modeling mixoplankton also comes with a series of challenges, and compared to the approach that Ken just showed us, this approach is more complex. We have more parameters. So there are some draw sides on this approach as well. So for example, the second point here, we can have very complex dynamics due to feedback processes, and many free parameters cannot help us to interpret model results and to constrain also these values. And other limitation is that we don't have data on mixoplankton activity to validate our predictions. So we could use biomass data from this fixed station, but since we couldn't validate the plot that I showed you, how much phototroph or the relative phototrophic growth of mixotrophs is, right? We might have overestimated mixoplankton biomass. So this is usually a limitation that is present in any modeling study regarding mixotrophs. And even though we were interested on these different flavors or strategies among mixoplankton, we still could only simulate a few functional types. So we have a limited capacity to simulate their diverse strategies, and especially capturing this full spectrum that we see among mixotrophs, from mainly phototrophs to mainly phygotrophs. And finally, who works with these models which have many functional types also know that it's very difficult to represent the coexistence of different strategies, and this is also the case here. So sometimes it's difficult to make mixoplankton coexist with their strict autotrophs or heterotrophs competitors. So this can also both underestimate or overestimate their impact on ecosystem functioning. So I think that's what I had to share now. I'm very excited to our discussion in a bit, so thank you for listening.
Thank you so much, Suzana. Another virtual round of applause. really appreciate such a lovely accounting for these different types of mixotrophs, binning them into some key categories, and then especially being able to validate that with data is really exciting. So remember, we're going to have a panel style Q&A for all three of our speakers at the end of our third talk. So jot down your questions in the chat or get ready to ask them in a few minutes. So we've heard from Ken and Suzana about different flavors of modeling mixotrophs. So how do we scale from these models to a global scale accounting of mixotrophy? So for our last talk, I'd like to turn the floor over to Dr. Stephanie Dutkiewicz, who is going to, who's based at MIT and who's going to take us up to the scale of the entire globe by integrating mixotrophs into global ocean models. So Steph, when you're ready, take it away.
Okay, and see my screen?
Yes.
Great. Okay, well, thank you very much. Before I get going much further, I just want to acknowledge my colleagues who've either helped develop the model or who've been part of projects to try and I use this model to ask specific questions. So as Holly just mentioned, we're sort of scaling up to the global, although I think Ken already did. But anyway, we're talking about large-scale models which have the physics, the three-dimensional physics, have complex biogeochemistry, so multiple limiting nutrients. And in particular, we've been interested in trying to model complex plankton communities with the particular interest in being able to ask questions about what sets planktonic community structure and diversity. And so the one of the models that which I'll be showing results from is a model that is designed like this with different functional groups. So for instance, diatoms, coccolithophores, and different sizes. So on the y-axis is different size classes of phytoplankton. And we didn't design this model specifically to look at mixotrophy, but we did put in some dinoflagellates and we made them mixotrophic. And so remember that as I talk further along, that we're really only capturing a very small amount of the mixotrophs, which we are aware of. We haven't given mixotrophic capacities to some things that possibly do have mixotrophy in them, which is coccolithophores. Anyway, well, we'll consider this. And the arrows here show which plankton are eating which plankton, and you can see that it really is quite an intricate food web. So how do we parameterize mixotrophy? And we are going to go even simpler than the simplest one that Ken indicated, and possibly relatively naively. Again, we were coming at this that we were trying to put different types of phytoplankton in, and that was not just mixotrophy. So the way we did mixotrophy is that the ones that are shown, are designed to be mixotrophic, they can photosynthesize and they can graze, and they do both at their maximum capacity. However, because we're particularly talking about dinoflagellates, they don't do either as well as specialists of the same size. So in these two graphs, the one on the left is phytoplankton maximum growth rates as a function of cell size, and on the right is maximum grazing rates, again, as a function of size. And the small symbols are lab results, and the different colors are different groupings. So for instance, the red are diatoms, the dark blue are coccolithophores, and the purple are dinoflagellates. And so from this, you can from these graphs, you can see that in the lab results suggest that dinoflagellates, and again, I'm not separating here which are the mixotrophic versus autotrophic versus heterotrophic, so there is some fuzziness in this. But in general, the dinoflagellates do less well in both photosynthesizing and in grazing. So we put that simple setup into our model and we run it out. And what you're looking at now is the annual mean biomass of all these different plankton types that we have in our model. And you can see that the mixotrophic dinoflagellates have very distinct patterns. There are a lot of them. They sometimes dominate in some locations. And so let's just, and I think Suzana already did a nice job of talking about the difficulties of being able to evaluate these sort of models in general. So some of the ways we have tried to do model validation is we've looked at data compilations, and the top one is from MERDAT, which collected observations from all over the globe, did the very difficult and relatively very uncertain thing of trying to take all that data and put it in the same units. In this particular case, it was carbon biomass. And that's an incredible task, and one that is very helpful for modelers who do model with carbon biomass as being our main currency. But you'll notice there's a kind of a stark lack of any, they did not include dinoflagellates in their data set, in their data compilations. We've also looked, we can also look at individual cruises. So for instance, the AMT has measured biomass of different phytoplankton types, and we can compare our model to those. And in particular, the importance of this is to be able to see how our dinoflagellates, or mixotrophs in this case, compare relative to other phytoplankton. So what is their biogeography relative to other types? Another thing we can do is to look at the timing of, for instance, the dinoflagellates blooms. So on the left is this on the bottom. Left is some observations from CPR, this is by Andrew Barton, and looking at the timing of the blooms of, for instance, the blue being diatoms and the black being dinoflagellates. And then we can compare our model and see, do we get the sort of separation of the timing? I think we can talk about when we go into breakout groups is new genomic data that's coming through is going to be incredibly useful, but it is very difficult to use at the moment because we don't know how to compare the omics data coming in relative abundances against something like carbon biomass, which we use in the model. And then the incredible importance of new targeted data sets, for instance, the work that Suzana is doing. And one thing in particular, and I really appreciate that from work, is the presence absence. It's not always that people document absences of something, and yet that it might be one of the crucial pieces to understanding biogeography, or at least in helping models in biogeography. So yeah, so we're sparse data, and that certainly is something we can talk about when this is the question at the bottom is one of the questions that we will be talking about in the breakouts. So just wanting to sort of finish off the talk with just, so we've developed these models, but what do we use them for? How can they be useful? And I'm going to give two little vignettes, neither of which are fully polished, but it can give you an idea of how we can use models like this to explore, in this case, mixotrophy. So what is the biogeochemical and ecological function of each of the types of plankton that we've put in the model? We can use a model like this to sort of do those sort of experiments. For instance, what happens if we remove the smallest phytoplankton? So this would be a sort of Prochorococcus analog. And how would that impact, for instance, primary production? So because we have the model, we can do that. We can run it everything exactly the same, just picking out that single phytoplankton and saying what happens. And you can see that it makes quite a big difference. This is the percentage on the bottom. It's a percentage change in primary production when we've removed the smallest phytoplankton, then run this model back out and then compared it to the run with the smallest phytoplankton. And these colors are actually saturated. In some regions, there's about a 20% change in primary production between these two simulations. Well, how about if we run another simulation where we keep, again, everything the same, except we remove the smallest diatom? You can see some other very interesting patterns, and there's a long explanations for why these patterns are the way they are. So really interesting to go have a look at the paper. But for this talk, what was particularly and really quite surprising to me is that when we removed the smallest mixotroph, the response was very muted. You know, it's in the order, I mean, it's not nothing. It's there is sort of 2 to 3% changes, but it was much lower than with other plankton. And we had to, you know, there was a lot of query as to why. When we delved really deeply into it, we found that when we removed most of the other types of phytoplankton, so like the smallest pico or the smallest diatom, the thing that took its place it only took, well, the thing that filled in for that niche was usually a phytoplankton of the same function, but of a different size. And this had a fairly then large effect because it changes the growth rates, it changes the trophic strategies. But when we removed the smallest mixotroph, it was actually a similar size copolithophore that filled its niche and a similar size zooplankton that filled its niche. And if you remember, the way we developed this mixotrophic dinoflagellate is didn't photosynthesize or graze as fast as its similar size specialist. And so the sort of query, and this is, I'm sort of throwing this out as something we can hopefully discuss, is if they're less good, and again, I understand that that's not necessarily true for all mixotrophs, but in this case, these dinoflagellates, if they're less good at doing something than their specialists, their roles can be filled at least as well if they weren't, for instance, extinct, as this experiment suggested. That's not to say that the mixotrophs are unimportant. They do, I mean, we've seen from the model that they really do fill, they can dominate in some regions. Their combination allows them to do really well. But I've really been struggling a little bit with these results because they may potentially, there's some subtleties of how these differ to, for instance, the work by Ben Ward in showing this increase in trophic transfer and organism size, and the work that Suzana just talked about when she removed the mixotrophs. And so the way I've been sort of wrestling it is the experiments I did actually are quite different, at least relative to Ben's one, where he had two different experiments. One where there was 2 guilds, so you could either be an autotroph or a zooplankton, and there were no mixotrophs, or where everything could potentially be mixotrophic. And here we had this very complex autotrophy structure in our model, and just wondering if that could explain some of these differences. So the mixotrophy is really important for the ability of the mixotrophs to have a really interesting niche, but it did not necessarily change the community productivity. And so I'm going to turn the question around: why have mixotrophy in a system if you could in fact have the productivity being similar with just specialists? And that's just to try and play devil's advocate. But just to finish off, and sorry, Carly, I'm going to be the one who goes over and everyone else is just to look at the implication of temperature functions on the phagophototrophy. And the prospect here is that there are some studies that suggest that phytoplankton may have a much lower Q10 than zooplankton. So here just showing grazing or phototrophy as a function of temperature. And you can see these different curves, the 2.7 being what has been suggested for zooplankton and the 1.88 phytoplankton, though there are certainly studies that suggest these might not be the perfect temperatures. But models in general assume the same Q10 for both phototrophs and for zooplankton, although there are a few models that suggest there may be some more, that they may be different ones. But then what do you do for a mixotroph? Because it's doing both. So is it 2.7? Is it 1.88? And another motivation for this study is work out of Holly Muller's lab, where it suggested that if you took a mixotroph and Holly, you'll have to tell people which one it was. And here I'm just showing the growth rate of this mixotroph. The dashed line is the thermal response curve of a control lineage, so the ancestor. And then the blue and the red are the same, the types that they've been allowed to several hundred generations of evolving in those different temperatures. And you can see how they've flattened that response curve. They've tried to bring themselves back to the temperature, to the growth rate that they had at their old temperature. And so this is just a very quick little show of some work together with Kevin Archibald, who's a postdoc with Holly Muller, who's leading this work, and also with Charlotte Love-Hutter. And the question is what should we be using for mixotropes in terms of their Q10, and what would happen if they evolved to have a different Q10 in a future warmer world? And so we run two experiments, one where the mixotropes have the same Q10 for both grazing and for heterotrophy, and the other one where they have a higher for grazing. And not surprisingly, it makes quite a big difference. So on the left is the experiment where they have the same Q10, and on the right where they have a different Q10, and sorry, in the middle, and then on the right is the difference. And you can see that in colder water, the mixotropes are highly, when they have a different Q10, a higher Q10 for grazing, they do much better relative to, they do much better and at the expense of the zooplankton, and the opposite in warmer water. So just to finish, and I am late, sorry, what is too simple? Remembering that global models on such as the ones I'm talking about are not focusing on mixotrophy. They also have to include the rest of the the competition with other zooplankton. The real query about how we're going to actually evaluate these models is for sparsity of data. This conundrum of what the functional role of mixotrophy is, sort of how my results sort of cloud a little bit some of these other results we've seen before. And then I'm interested of just how we design these models in terms of things like temperature, but we could also think about different things of light and nutrients, etc. And thank you. Sorry, Holly. Finish there.
No need to apologize. It was worth every minute. Thank you, Steph. And we'll do a virtual round of applause for you, and thank you to all three of our speakers for such excellent and stimulating talks. I'm sure that all of you, like me, have a lot of questions for the speakers about modeling of mixotrophy. So I'm going to hand it off to Jessica, who will moderate the panel discussion.
All right.
Sorry.
Thanks, Holly.
I can't actually, we can stop sharing. Sorry, apologies.
No, that was fantastic. So we have about 8 or so minutes for some questions, and feel free to put questions into the chat or raise your hand. And I could just start us off if you would like. This is a question directed to Ken, but actually to anyone, about including the cost of space. Any amount of space taken up by machinery for phagotrophy or kleptoplasty will reduce space for other things, such as storage. Have you considered ways to conceptualize that trade-off? Ken, you're muted, sorry.
Yeah, sorry. The model I produced here was really the minimal one. It doesn't include any trade-off. Everybody can do everything. But we have actually included those trade-offs. So we have made the next more complicated version, for we say they can invest in phagotrophy, phototrophy, and nutrient harvesting. And that specifically has the trade-off of that it takes essentially space or biomass away if you invest in one function or the other. And then you can look on the given condition, what is the optimal strategy? So that's kind of the next level there. I'll put a link in the chat to those two studies where we did that. But qualitatively, you see similar patterns. You just get a little bit more of the, say, the specialist generalist spectrum that Steph talked about represented.
Great, thanks. Any other questions? Feel free to put questions into the chat or I'm not sure if there's a raise hand function, or maybe just speak up.
So one question that I had, and I guess this is addressed to all three speakers, is it seems like each of you had to reckon in different ways with the challenge of mixotrophs either being not as competitive as specialists or being too competitive. And that produced issues with coexistence, I know, for Suzana, and also this interesting phenomenon for Steph with niche getting sort of filled by someone else. So how did you kind of reckon with that calibration, and what sorts of empirical data might help to constrain that better? Because it seems like there could be many ways to try to control that.
Do you want to go first, Steph, or I go?
Well, so for us, it was this assumption that they couldn't do either thing. They couldn't be graze or photosynthesize as fast as as other things, and it was how much that is reduced makes a difference, and that's kind of one of those unknown parameters, and maybe that would be something to know, how much that cost associated with having both would be something really useful. So, for instance, if I dial down the amount that the mixotrophs graze, I can kill them off, or I can make them super bugs. And so I think something like that. What is the cost of having both? And I'm assuming that actually would be very different depending on whether they're constitutive, not constitutive. I mean, I think that's there is a wide range on that.
Yeah, I think I had a similar struggle. For me, it's very like sudden. So if you make them a little bit better, the mixotrophs, they will dominate and the others will die. Or if you do the opposite, is also very sharp, this transition in coexistence. So I agree with Steph that we are probably missing some cost to better parameterize these. But when I think about data like that could help, for example, on my case that I was looking at a fixed station rate, maybe something that I would like to have done going to the past is actually using some techniques that can, we can incubate the in situ community, for example, with a tracer, and then we can mark the feeding vacuums, and we kind of have estimate a percentage, right, of how much of these nanoflagellates, for example, are autotrophs, or how much are mixotrophs and how much are heterotrophs, just based on chlorophyll and feeding vacuum markers. So this is something that if I had like over the year, I would be very curious to see and see if it could help model validation. But still, we would need to do something in the model construction, as Steph was saying, to be able to better represent this coexistence.
Yeah, if I have a comment on the coexistence, I mean, this is the eternal problem. When you're modeling types which are fairly similar, any kind of food web model has that, and the size-based models have it to some degree, but not as strongly as the other ones because they're fairly different. But it's also a very primitive model, so it's not to say that they're better. And the only way to get that coexistence stabilized is to have a density-dependent effect, that is some process that depends on the biomass, and that's tricky. And usually all biogeochemical modelers know what they do. They have a quadratic term, so mortality that essentially is linearly with the biomass. And we also have that. That is some kind of representation of viral attacks. The only way we are validating or calibrating that is to make sure that this is a small mortality. We don't want it to dominate over the predation mortality or anything else. And yeah, but coexistence is an internal problem. It's not something that goes away easily, and adding extra trade-offs, I mean, that's always what we think, but it really doesn't solve the problem. The problem is only solved by density dependence. That's how it is.
Sorry, Ken.
That's it.
I mean, the other thing is also different substrate. I mean, so allowing you can kill off a diatom if there isn't any silica, and so that would allow some additional mixed. So I think also adding in that in some ways the global model with all of the the iron and the silica and nitrogen and phosphorus and diazotrophs has a little bit easier time keeping coexistence just because of the multitude of other things that can set it.
Absolutely, and it also has lateral mixing, which also helps.
All right. Well, if there aren't any other questions, then we can just give the charge for the breakout sessions. Is that okay, Holly?
Yeah, absolutely. So I'm pasting into the chat now the prompts for the breakout room discussions. So we'll take 20 minutes in small groups. Each of the groups will have someone from OCB leadership there. So we'll be taking notes in this Google document. So we'll take 20 minutes to discuss these questions, and then we'll come back and briefly debrief and talk about plans for the upcoming OCB Mixotropes and Mixotrophy meeting in November. But essentially, we'd like you to think about this dynamic tension between being a mixotroph and being specialized, what sorts of empirical data we could leverage in designing and improving mathematical models for mixotrophy. And also, as modelers, we're challenged to ask what are the key traits and key aspects of an organism that we put into our equations. So what are the most important attributes of mixotrophs that we need to include in our models? So with that, I will start the breakout sessions, and you should get an invitation to join your breakout room. Enjoy your conversation. Welcome back, everyone. We hope you had a great discussion. So as we wrap up in the last few minutes, I'll just invite the folks who were facilitating the breakout rooms to share perhaps one key point that stood out to them from their conversation. And I can start. So in breakout room 3, or actually I don't know what breakout room number we were in, we had a really great discussion that, as usual, veered into all the complexities and challenges of studying mixotrophy empirically and some of the limitations. But we left feeling inspired by the interplay between mathematical models and empirical data, and especially as we seek a better mechanistic understanding of how mixotrophs turn on and off different aspects of their physiology, and we look for empirical data to help us define those mechanisms, models can be really powerful as a way of testing different hypotheses for those, and can also provide us with inspiration for the next experimental questions to ask. So Jessica, do you want to share a highlight from your breakout room discussion?
Sure, though I don't know if I can just pick one. One of the things that I thought, so we spent a little bit of time talking about at the beginning, was this choice of a temperature response and how we can know what the mixotrophic temperature response should be, and actually even whether there is sufficient evidence to suggest that there is a distinction between the phytoplankton and zooplankton temperature response, and that being kind of a key unknown in the field. But kind of moving on, talking a little bit about the limitations of data, not only identifying who can be mixotrophic, but who who are mixotrophs or are capable of being mixotrophic, but when and where that actually happens, and the limitations of data that actually get us there. And finally, there was just a overall, very short discussion on how we choose which organisms within models to be mixotrophic. And yeah, overall, I just found it to be a very lively discussion.
Nicole, do you want to share highlights from your breakout room, or Karen?
Sure. So we discussed a variety of things, but by the end, one thing we kept coming back to was this idea of assimilation by the mixotrophs. So from their grazing, what are they actually assimilating into their cells? What are they really taking away from it? And because that could help in models with a lot of, with biogeochemistry models, and just really starting to, look at that. Are they breaking down amino acids? Are they going down to more base level elements? Are they respiring most of the carbon? Are they just using that for respiration? Are they incorporating that carbon into their cells? And then what kind of controls do you have on the like what's being assimilated and being assimilated from phototrophy versus phagotrophy and things like various things like that. I think there's a lot of room to expand that discussion, but it was the start of something I think people kind of got a little bit excited about by the end.
Terrific. Great. Well, so we will generate a summary document of the discussion, and we'll also be posting a recording of the excellent talks from today's session if you want to review that or pass it along to colleagues. As we wrap up the meeting, I want to again thank so much our three speakers for your excellent talks today. So thank you, Ken, Suzana, Stephanie. Really appreciate it. And as I think we discovered in the breakout room discussions, there's lots to discuss and lots that fascinate us, and also a lot of challenges with studying mixotrophy and incorporating our empirical results into models. So in November, when we have our next OCV mixotrophs and mixotrophy working group session, we will not only update you from some of the work that the formal working group members do in the October meeting, but we'll also solicit your input for the upcoming year of these meetings. So what are the topics that keep coming up again and again that we should dedicate one of these 2-month sessions to? What are some of the themes that you're excited to explore more about in our working group? Anything you'd like to add to that, Nicole, Jessica, Karen?
Just a reminder, we've got a session for ocean sciences. If people would like to submit abstracts to, they're due on the 29th of September. Hoping to have a lively session there as well. And then to wrap up by thanking Holly for moderating. She kept us on time, so well done.
And thank you, Jessica, also my co-organizer for the session. So hats off to you.
All right, we'll see a lot of you for the October meeting, and for everyone else, we will see you in November.
Thank you, everyone. Have a great rest
Bye.
of your day.
Thank you.
Thank you. Bye.
Bye.